Stanford University.
So I teach statistics and data mining, and I use R, and I actually used R since '97. So I started using R a long time ago, before we had enough data. At the time we didn't have enough data, now we have too much data. And I have the same slide as Hashem had before. My roadmap is about the same as what he had up there. But it shows you basically that there are many entries and reasons for using R, and the talk before me was a wonderful example. He actually made all the plots with tools I'll talk about, and all the metrics and analysis was done using R. So I thank him very much because he set me up. But so it's very complicated to explain using every part of R, and I'll try and give you the aspects that I use the most in my own work. And here are some of the advantages. From a scientific viewpoint, reproducibility is essential, and nowadays when we have these large simulation models for climate control and biological studies, it's very important for even the simulations to be reproducible. So you have to have a way of communicating scripts and reusing what you did before, and those tools exist. And so R is an open source project, so all the code is open source, and anybody can reproduce experiments or things that we do, simulations, analyze data if the data is open. I use R for prototyping. So it's very often the case that somebody comes with me to me with some data, and it's a prospective study, and they want to know what's the best method to analyze this data. And so what we need to do to start with, before you have large amounts of data in a smallish study, usually when it's images, it can still be a couple of gigabytes, but we'll do a prototype study. And these prototype studies are very easy in R, because there are almost 4,000 packages, statistical packages. Any model that was written by somebody who does statistics will probably have somebody else or themselves who wrote a package to do it. So we'll see that the many different types of data, like mass spectroscopy, DNA microarrays, sequences, there are packages for calibration, normalization, quality control, for all the different types of data. So prototyping is extremely efficient in R, and data input and cleaning, filtering, data imputation, things like that. The plots that you saw in the previous slideshow were made with a very nice tool, which is the best visualization package available, which is called ggplot2. And those were actually built on a system which was very thought through. Over 15 years of work, Lee Wilkinson developed something called the grammar of graphics, and its way of layering plots to have more and more information and to be able to make them and remake them very, very quickly. So again, it's visualization and reproducibility. So you don't want to have to repeat everything from scratch. You have the script available. And so again, you have an open source community, so you can ask questions, and you have documentation. So those are some of the advantages. I'd say that some of the difficulties that I encounter in my work, I don't have petabytes of data. I teach students how to use methods that they go forward and often use very large amounts of data, but it's still, the main drawback is, by default, R takes the data and puts it in memory. And so there are constant workarounds for that. There are addressing problems even if you have a larger amount of memory. We're working, many people are working to try and counter that, and John Chambers, who's here, he'll be here for the after workshop meeting as well, And he told me today that the R core, who work together as a team, that's one of the main things that they're working to address. But in the meantime, there are actually quite a few workarounds, and I'll talk about that. One of the other problems is because it's an open source project and you have 4,000 packages out there, you had 4,000 different teams of people who wrote documentation and code, and some are better coders than others, and so it's very unequal what you have. And you get what you pay for, but the advantage is it's open source, so you can make it better. But you do have to pay attention to that, and a lot of profiling has to be done. And actually now, there are businesses which are built up on the idea that you may need to have something which have higher quality control, and there's a commercial enterprise to make some of these tools available commercially. The other difficulty with R is the same as the advantage, that is, it's a community of users, and if you're on the R help list and so on, you'll see that some of them are quite nasty. In the sense that statisticians consider themselves very self-righteous, and they're always telling the other people that they don't know what they're talking about, that they use the wrong vocabulary, that they're, and that's a common theme. And so there are the kings or the emperors of R, who some of them could do with a little bit of PR, especially because they're over the other side of the pond, as they'd say, and so they come from the same place I do. They're a little bit more outspoken. But as a whole, it is an enormous community. So you find yourself with a number of users, which is so large that if you have a problem and you're trying to solve it, all you have to do is type it into Google. And somebody else will have had the problem before you. And if they haven't, then you can ask the question. But it's an advantage and a disadvantage. So what I've found is it's the best design platform for any kind of statistical analysis. And what I mean by that is somebody will come in the door with some data which it comes from microarrays or some images, and they'll ask me to analyze the data or think forward, how could we write a grant which would analyze this data well? And you can take off-the-shelf packages which can take data in very structured, complicated formats that come off strange machines and actually compare the output. So I did that, for instance, in a study I was doing on image segmentation for counting cells. We were trying to compare 17 different types of statistical learning algorithms, and all 17 had various package or implementations in R, and we could just compare them and decide which one did the best. And although I had a favorite method that I really wanted to use, the student proved to me that random forest was much, much better. And I don't like random forest because it's more of a black box, but in this case it killed the other methods. It had 98% accuracy, so we went with random forest. And we didn't have to do very much work before making that decision. We then made a standalone application, which is actually open source and written in Java, which is used by the doctors and which stands alone and doesn't use R, because that's another thing which is a disadvantage with R, is the learning curve. So you're not going to be able to tell a trained pathologist, you have to learn this command line language and be able to interact with your data. That just doesn't work. So, but it's very good at prototyping and profiling. So there are various tools which enable you to see where the bottlenecks in a program are, in an algorithm, and you can take those parts out, and because R is interpreted, you can take those parts out and make them pre-compiled and interact with connections to C, Fortran, Python, anything you like, Ruby, all of those exist already. So as I said, I use it also for teaching, and I teach data mining. And we start with doing many, many different examples, smaller, and then we scale up when we're teaching. And for instance, I use a a competition type setup for teaching data mining, and we used Kaggle, which is a nice, which was inspired in some sense from the Netflix example, where you can get people very interested in perfecting their algorithms if you put out data freely. And so to the people who come with large amounts of data, like the previous speaker, but the other companies who were talking this morning, I'd say, why don't you think about taking some of your data and making it publicly available, even if you have to anonymize it, because it would be very nice to have more open source data as learning tools. That's definitely something in the realm of large data sets which is missing, although recently NASA put up a Kaggle competition on image analysis with data. Unfortunately, it wasn't real data, it was all lensing simulated data, but having a lot of data to play around with it would be a very nice teaching tool. So, as I was saying, you can interface R with many different programs, and John Chambers, who was at the origin of the first program, which was called S, which is the ancestor of R, who will be around. He has a project called the Omega Hat project, which has many, many different interfaces available. So you can interface R with Firefox, with Google Docs, with Google Maps, with JavaScript, with Amazon. All of those exist already, so there are bridges between R and anything else that you'd like to see. So I made a link. These slides are going to be available, and so most of these are already alive. So here's a link. So this is the project, and you can see all the different possible interfaces that they work on and how you can sort of harness these bridges which exist. So that's something very nice because it's not an isolated system. The thing I use it the most for is, because I do mostly sort of bioinformatics and biology-based problems, is I need access to very many standard databases. And R, through particular the Bioconductor project, which maintains all these interfaces and keeps them up to date. There are packages such as Annotation DBI, which has access, you have access to GenBank, to the Gene Ontology, to Unigene. So you have these bridges which exist already to the databases, you just collect the data. And that's extremely important in what we do because we have such heterogeneous sources of information that we need to put together. The other thing that one remembers is that statisticians actually spend, I tell my students it's 80%, probably I'm lying because I still want to have students, it's probably 90% cleaning up your data. And when sometimes I show students that I'm doing this, I'm cleaning up the data and I'm filtering and I'm taking out missing data, and they say to me, don't you pay people to do that? And most of the time, actually, no, you don't, because that's where all the information is actually going into your brain. And a lot of the slicing and cutting up into pieces can be done through R. Although if you have a very large database, it's again, the workflow I use is I try a part of the data, I see what needs to be done, and then that gets reprogrammed in another language. So it's again a case where we very often prototype in R, and then we have it reprogrammed so that it's more efficient. But we never know what to do when we start off. Where are the missing values? Where are the problems going to be? The other thing that we can do, and especially if you have very large data, statisticians like to use sampling and resampling of the data, and they call resampling the bootstrap, but it's not the bootstrap in the sense that computer science think of the bootstrap, but it's definitely something that is very important. So random number generation and having tools which allow to do that in a very sure way is definitely very useful. I can't list all the possible ways that are available already for doing missing data imputation. So as you know, if you look at data like Netflix data and things like that, there is a large proportion of the data missing, and the way you fill in the holes actually will have an influence on what you find. So you have to do that in a reproducible way. When I mean reproducible is you're going to choose a certain threshold. You'll say you'll replace 20%, so on, or you'll reject the other ones. But that threshold, you want to be able to replace it very easily, and that's why the reproducibility of R is so useful. So Sweave or various tools which now exist allow you to do that many times automatically, changing the threshold. So these are lists, and they're all the different types of data imputation. The data I use all comes off machines. So they come off machines, but it's mostly not the internet. The machines they come off are sort of mass spectroscopy machines, microarray machines, various types of sequencing machines, a lot of images, medical images, but also videos and things like that. And this data comes in always some nasty format. They don't come out of the machine in Excel, or they're highly structured and very complicated, and it's always machine dependent. So if every time a data set came in like that, I had to program exactly that format. And I'm remembering the Agilent. I was the first to analyze some Agilent data, and I had to actually write the program that did that. It just is a complete waste of time as far as I'm concerned. And so that's another thing which is really nice. That is, all those formats, there are packages which allow you to access them. So for the ordinary type of import from databases, there are the HDF5, DBI, foreign, GData. These are all names of packages which already exist. The first time I had some really large data that I couldn't keep in memory, I used NCDF, which is a standard way of hashing the file so that you just have the addresses, and you can then have slices of the data in memory. You don't have everything at once. And now there are much better ways of doing that. But we have interactions with all the different types of databases, and including Excel. Image formats, so medical image formats, which are standard. So there are some that come microscopic images. There are a package called EB image, MRI images. Tractor is a project which is an R4 project, which combines all the different types of MRI imaging. DCOM is another one. For microarrays, there has been so much interest in the bioinformatics world that there's a separate project within R, and I'd say that if I was a physicist, I'd say we'd do the same as what the people doing bioinformatics did, and do a version of bioconductor, but for physicists. Because in fact, what bioconductor provides is all the quality control, and it's updated, and it has much stronger documentation requirements than the standard R, which you can get off CRAN, but the documentations are often not standardized. So you can do all kinds of nice graphics and input very easily. And for mass spectroscopy, there's a whole, on the R website, you'll find there's chemical and physics tasks, and you'll see all the different formats for mass spectroscopy. And it's not just read in the data as it comes off the machine, but it's also all the different technologies, for instance, wavelets, or the different methods for actually analyzing and finding the features in the mass spec data, but also interfacing it with various databases so that you can find out what the compounds are and things like that very easy. So the plotting feature, although it might seem crazy to want to plot terabytes of data, it's still extremely important to keep in touch with what's going on in the data by looking at it, even if you have to take slices of it. And when I say what's going on, it's not just in one dimension. The kind of data that we have is not just homogeneous reams of data of just real numbers. What we have is very often data which we have a network or a tree which connects all the possible species, say, and then we have counts of presence of the species. And I work, for instance, on bacteria in the gut, and you have thousands of different species, but we make measurements of millions and millions of sequence reads. But you have to put that together with the clinical variables, and you might have biopsy data. So we have a source of data which is extremely heterogeneous. And you want to try and combine all of that to get a picture. And so these types of plots are very standard. This one was made by a package called ADE4, which allows you to do high-powered multivariate plots, but which give you very much information. This idea of layering plots and doing facets and improving them so you can see many things at once outlined in Lee Wilkinson's book, and the implementation is ggplot2, and I'd recommend there's a Google talk by Hadley Wickham, who's a young rising statistician who's written a lot of very nice programs. Some of them have a defect, that is, they can be slow, and although they're extremely aesthetic and pleasing, it can be frustrating, and it would be nice to be able to find better ways of doing these faster. If you have very large problems, there are different motivations for the type of high-performance computing you need to do on them. So it might be that you want to, that parallelization is enough. That is, all you need to do is parallelize your program, and then you can use many cores, and that's possible through, here are a few examples of packages. So there's RMPI. Snow was a very early one by Luke Cherney, and then there was Snowfall. That's another one. There are packages which have, in fact, implicit parallelism, and multi-core is a favorite. And there's a package called MapReduce, which follows the MapReduce approach, although it's not as powerful as the ordinary one. So because the data don't fit in RAM, here are some solutions to that. And I'm not saying that they're all perfect, but some of them work quite well. And so there's RHIPE, so RIPE, and FF. Big memory is one which allows you not to have all the data in memory and just uses a large amount of indexing and hashing. And so there are some examples, and I'll point you, because of time, to an example of managing large data sets, which was presented using FF. So there are a couple of those that you can point on. And so there are some specific out-of-memory solutions to some of the models that statisticians like the best. So the most common command, or the one that statisticians use the most, is LM. It means linear model. And it means if you do any kind of regression where you have on the left a Y, which is a response that you're trying to explain, then you'll want to use big LM, which is developed by Tom Lumley and co-authors, and then there are other packages which also are specific to certain types of model. And then now, as we see the adoption of Hadoop, there's been a definite push by several teams, and Hadoop streaming is one of them, which allows you to use MapReduce scripts. And there's another one, there are two others. The Revolution has something they call the RevoScaler, which is a proprietary package which you have to pay for, so I won't talk too much about that. Ricardo is a project which is being worked on by people at IBM at UC Santa Barbara. And this has the idea of using this destructure the big data for certain problems, and then work on the parts that you can where you need to do, for instance, vector or matrix analysis. And so I just wanted to finish by saying here are the problems that I know are unsolved, which might be interesting. We very often use methods which you do, for instance, nearest neighbors. In the Kaggle competition I ran for my students, it's very often nearest neighbors wins. And so for all kinds of complex distances, if you have too many points, your distance matrices get completely out of hand. Another hard problem that we can't address is, of course, when you're trying to do extreme values. And the dynamic programming, the speed at which you can do these nice ggplot problems are definitely something which is a barrier. The last thing I just say is that we still don't know how to deal with data which is not homogeneous. So we have heterogeneous data. And this is just an example where we have a network of genes, we have microarrays, we have images, and we need to combine all of that together. Now, if you have a very simple indexing structure, that's practically impossible to do. So the heterogeneous approach is definitely one of the most challenging, I'd say. So the slides have all the links to all the information. And so I'd like to thank all the people who've developed this open source code, and in particular the bioconductor group, who put a lot of work into making things very dependable. And I have a PhD student who I work with on large data sets, Nelson Ray, who's very helpful and also helped me with my big data mining class. Okay. That's all.
We have time for about one question, if there are any. Yes, there's one.
So you're talking about large data, and I'm not seeing a lot about parallelism in terms of like what we do classically under MPP. I mean, because we have machines which have 300 terabytes of memory. Surely your data is not that large.
No, I understand. That's the problem isn't there. It's in the addressing of the data within R. It's not exactly, it's not the problem of the data. It's just the structure of the way that R is written. So it doesn't deal, you can't use that amount of data without a certain amount of work.
Well, but I know of different parallel R packages. There's RMPI, parallel
yeah.
R.
All of those parallel packages allow you to, of course, to treat them together. But it's the problem of how you aggregate afterwards. There's no standard model that works today for doing that. So what I showed you is all the different projects that different people prefer. And it really has to do, when you say parallel, it's very easy to parallelize certain computations because you don't care about what's going on in the different pieces. When you're looking for extreme values, you have to have much more communication between the parts, and so that doesn't work.
Okay, thank you very much. Oh, okay, if it's a quick one, okay.
Just a quick point that you could use the server-side PG-R in Postgres. So you could create, for example, aggregates using R functions. And it works pretty well for terabyte-sized data.
Right, okay, thank you.
All right, thank you.
For more, please visit us at stanford.edu.